Over the last 4 years, we've been developing our trait and technology pipeline, which is now fully delivering traits to be launched to the market. As a first step in this pipeline, we work on gathering and developing genetic resources.
Our trait discovery team focuses on developing groundbreaking novel traits in wild species and making them available to our commercial breeding programs. We perform several experiments to investigate our genetic resources for traits which are important for breeding, including resistances to pathogens such as fungi, bacteria, insects, and viruses. Our phytopathology specialists focus on the performance of bioassays to identify sources of resistance and to test them under the most harsh conditions so that we are able to make sure that our INTRINSA varieties can withstand the most harsh conditions. Our trait exploration specialists also focus on improving traits such as vase life, transportation resilience, heat and drought tolerance, plant architecture, flower morphology, multiplication rate, rooting ability, and future product forms. In many cases, the sources of resistance are not easily available for our commercial breeding programs. Therefore, one of our main activities is to develop and maintain a large scale of genetic resources, and with the help of our cell biology specialists, then we can exploit these true gems of nature. Artificial intelligence helps us in implementing image-based phenotyping in a large set of plants from our breeding programs. With the obtained information, we can support our global breeding programs by providing new insights into our genetic material. With this, they can make better crossing designs and better decisions that will allow us to have a more efficient, reliable, and faster breeding progress.
When such a novel trait is targeted by our trait exploration team, the trait genetics team comes into play. They study the heritability of the trait and develop molecular markers for effective breeding later on in the research pipeline.
We use genomic data to develop molecular tools to support the discovery team to screen large numbers of plants for the pathogen load. We detect whether a plant is being infected by a pathogen, how severe the infection is, and which pathogen is causing the infection. We offer our breeders molecular selection tools and resistance genes to predict whether a plant is resistant or susceptible to a specific pathogen. These tools are applied often as early as the seedling stage, which is more cost-effective and faster than performing greenhouse pathogen tests.
With more and more information becoming available about our crops in relation to the traits they harbor and their genetics, we open the possibility for floriculture to move into the era of predictive breeding.
We are developing tools to make recording phenotypes faster and more reliable. Phenotypes could be traits such as disease resistances, strength, yield, and even consumer preference and ornamental quality. By using deep learning, we extract meaningful phenotype information from large numbers of images. We predict the phenotypes of a plant long before we can observe it. And even better, we predict the phenotypes of possible offspring between plants, such that the breeders know which crosses to perform and which crosses they shouldn't perform. This modular approach means that we can apply technology from small to large crops and for traits influenced by a single gene, such as disease resistances, to traits influenced by thousands of genes, like strength and post-harvest quality. All of this information is available in a condensed and easy-to-use way.
With novel traits developed, we rely on cell biology and physiology specialists to allow access to the traits for our commercial crosses. Our scientists develop methods for fast breeding cycles and mass propagation to be applied once the targeted new varieties have been developed.
The major challenges and distinctions of breeding companies are how they generate, maintain, and exploit genetic variation. Here within cell biology, we help breeders to access the natural genetic variation which was beyond their reach without the help of technology. For instance, embryo rescue techniques are used to lift reproduction barriers within wild species and commercial cultivars to transfer resistant traits. Those have been lost during the domestication process. Also, ploidy alteration and double haploid technologies provide tools to breeders to stabilize and exploit genetic variation. Biological processes are used to shorten the plant life cycles by speeding up plant developmental programs. For instance, culture media with right amounts of nutrients and hormones and good light and temperature conditions can be used to significantly reduce the plant life cycle during the breeding program. Combination of faster culture conditions and faster molecular selection processes provide breeders the methods to fastly integrate novel traits within commercial breeding program and fastly launch the product to the market. By utilizing totipotent nature of plant cells, it is possible to regenerate whole plants from single cells. Large plant cell cultures provide amazing opportunities in plant culture. They allow mass production from smaller starting material with automation opportunities. Large-scale in vitro production processes also provide ultimate hygienic levels without the use of harmful chemicals.
Now having facilities to match our ambitions, we are ready. Ready to deliver new varieties with groundbreaking traits, which can make the floriculture industry flower sustainably. With innovations like powdery mildew and agrobacterium-resistant roses, rust-resistant chrysanthemums, powdery mildew-resistant calanchoes, and TMV-resistant petunias, we can safely say we are just scratching the surface. With over 70 active research programs and more than 50 traits under validation, we are ready.